Hello, the topic that I'm covering today is Chapter 10, which is sexual reproduction and meiosis. So, there is a whole type of cell cycle that is unique in order to produce eggs and sperm. Okay. And that specific type of cell cycle is called meiotic cell division or meiosis. All of your other cells are divided or replicated by the process called mitotic cell division or mitosis, which we learned in the previous chapter. This, what we're learning here, is only applying to making eggs and sperm. Let's review some of the terms that are applicable here. So the sexual life cycle of a human is made up of meiosis and fertilization. All of the cells in your body, other than eggs and sperm, so every cell, liver, skin, heart, brain, eye cell, all of those cells, they're called somatic cells. So that just means body cells that are doing your regular work in your body for everything that's necessary for you to survive. And they are all diploid. So all somatic cells have two sets of chromosomes. One full set of 23 that you got from the maternal side, and full set of 23 chromosomes that you got from your paternal side. So the egg and the sperm that made you each brought in 23 chromosomes, or one set of each. Gametes, on the other hand, eggs and sperm, it's like saying parents for mom and dad. It's gametes, one word that is for both eggs and sperm. They only have one set of chromosomes, so we call them haploid cells. So the terms somatic versus gametes are just what type of cell is it? Gametes are just the cells that are used for reproduction only. They don't do anything for your body other than that. They're eggs and sperm. Somatic cells are all of the other cells in your body, everything else other than eggs and sperm. The terms diploid and haploid are referring to how many sets of chromosomes are in these cells. Haploid cells have only one set of all chromosomes. Diploid cells all have two sets of chromosomes. Offspring inherit genetic material from two parents. How that happens is there is an egg that only has one set of chromosomes. In humans, that would be 23 entirely unique chromosomes. This cartoon only draws 3 for simplicity. And a sperm only has one set or each chromosome. 23 in humans. When the sperm fertilizes the egg, the DNA from the sperm fuses with the nucleus of the egg, and now you have a diploid cell. And this diploid single cell is called a zygote. So zygote is the term we use for a fertilized egg. Just one cell, but it is now diploid due to fertilization. Okay. So this maternal chromosome is homologous to this paternal chromosome. This maternal chromosome is homologous to this maternal. And the littlest ones are homologous to each other as well. So there are 3 pairs of homologous chromosomes in this example. In a human zygote, there would be 23 homologous chromosomes. And remember that homologous chromosomes, like this one and this one, they each carry information for the same traits, but they are not identical to each other. Eggs are much larger than sperm. They have all the necessary components for the cell survival, while sperm is a vehicle for DNA to be moved from one individual to another. So there's just a nucleus with the DNA, practically nothing else in there. Some mitochondria in the tail to keep it beating. When the fusion happens and the DNA from the sperm enters the egg, the tail and everything else generally fall off and get recycled. So life cycles of sexually reproducing organisms like humans involve alteration between this haploid and diploid stages. Okay, this is very different in different types of organisms. So plants, fungi, different kinds of protists, they have these very different ways to reproduce than we do. In most animals, the diploid state is what dominates, meaning that most of our life we are in this diploid state. Let's look at a diagram of this process. Okay. So we already talked about this haploid sperm and the haploid egg. Haploid is indicated by symbol N. They fertilize, go through fertilization, and produce a zygote. And it is now 2N, or diploid. It has two sets of chromosomes, right, with homologs for each. This zygote, this diploid cell, will continue to divide by mitosis, what we learned in the previous chapter, to create two cells out of the one zygote. Then the two cells will become 4, the 4 will become 8, the 8 will become 16, and so on, until you have a full baby. But before the baby is born, a few of the diploid cells from all this division will get put away in the gonads, in the developing gonads of the baby, and those cells are set aside from the beginning to eventually be used for making gametes. And these diploid cells that are put aside are called germline cells. They're not used for anything else but to just be a reserve for something that will be used to make gametes later. All of the other cells in the baby's body are called somatic cells, and they are diploid and do all the different jobs that our cells do. In order to become a developed adult, more cells have to replicate, so we become bigger. When humans reach reproductive age, puberty, the germline cells, those diploid cells that were put away from before you were born, they will now start to go through the special cell division process of meiosis in order to create eggs and sperm. It is a very important process because it takes a diploid cell, 2N cell, in humans that's 46 chromosomes, and it produces eggs or sperm which are haploid, 23 chromosomes. The only cell division process we have learned so far in chapter 9, mitotic cell division, it starts with a diploid cell in humans that's 46 chromosomes, and it results in 2 diploid daughter cells, each 46 chromosomes. If we use that type of cell division to create eggs and sperm, then each would have 46 chromosomes. When they fertilize each other, there would be 92, and then all the cells would have 92 chromosomes, and the next generation, they would produce eggs and sperm with 92, which would fertilize to become 184 chromosomes. So every generation, there would be a doubling in chromosomes. That's an evolutionary disaster. There's no way our cells could get used to that huge of an increase in the amount of DNA. I mean, it just does not work. So we need to have a zygote that returns to the normal 46 chromosome. And the only way to do that is if eggs and sperm are half of the diploid amount. And so for that, we need a special cell division process called meiosis. So the purpose of meiosis is to take a diploid germline cell and from it make genetically unique haploid gametes so that upon fertilization they can form a diploid zygote. In order to do this, in order to have half of the amount of chromosomes necessary, half of the amount of chromosomes than our regular somatic cells, we need to go through 2 rounds of division. So meiosis has 2 rounds that follow one right after the other, called meiosis 1 and meiosis 2. And each has prophase, prometaphase, metaphase, anaphase, and telophase. We don't describe the prometaphase because we just lump it with prophase. But the whole cycle, similar to what you learned in chapter 9, does happen. When you see any of these names, prophase, followed by number 1 or 2, you know that we're talking about meiosis. Okay, so we begin the process of meiosis, the special type of cell division that will make eggs and sperm at the end. We begin with a diploid cell. Specifically, it's a germline type of cell that was set aside from before you were born. It's a diploid cell, and that cell will go through all phases of interphase. So G1, S, and G2, just as we learned it before. All the same things happen during these stages. During S phase of interphase, of course, all the chromosomes duplicate and will be made from sister chromatids now. So you have, at the end of interphase, 92 chromatids inside of that cell. Meiosis 1 will be dividing them in half, 92 divided in half, 46. And then you will divide them in half immediately again. 46 divided in half will be 23. So there will be 2 rounds of division. Meiosis 1 is the first, meiosis 2 is the second. Each consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. Remember, prometaphase we're just lumping in with prophase. Okay. Again, meiotic cells go through the same interphase period and duplicate their DNA and create sister chromatids, just as we did in the previous chapter when we were talking about mitotic cell division. After this interphase, when you have 92 chromatids, these germline cells will enter meiosis 1, or the first round of division. More specifically, right after interphase, they enter into prophase 1. Prophase 1 is the most unique phase that differs the most from mitosis. Some things are the same as what we've learned in the previous chapter for mitosis. All the chromosomes that are now made up of sister chromatid pairs, they coil very tightly, as tight as they will get, and they become visible under the light microscope, just as we would have described prophase in the previous chapter. Nuclear envelope disappears and spindle forms. Spindle is that special cytoskeleton that is designed for the division of chromosomes. And so far, both of these 2 bullet points are the same as what has happened in mitotic prophase. However, there is a very unique feature in prophase 1 of meiosis, and that is a process called synapsis. So first, homologous chromosomes, which are made up of two sisters each, find each other and stick to each other with proteins that act like Velcro to become tetrads. So let's see it. So this is your, let's say, maternal chromosome number 2, and its identical sister chromatid. These two sisters are identical, and they were made in the S phase of the interphase. And this at the bottom here is chromosome number 2 that is paternal and its identical sister chromatid. Remember that homologous chromosomes are not identical to each other, but they are the same size and carry information for the same traits. In mitosis, which was what we learned last chapter, homologous chromosomes never connected and had nothing to do with each other. They were acting completely independently. Both were made out of sister chromatids, but they had nothing to do with the homologs. However, in prophase 1 of meiotic cell division, of meiosis 1, homologous chromosomes find each other and come together with the use of these proteins that hold them. So there are 4 sisters that are held together temporarily. In a human cell, this means that we have 23 tetrads in the cell. And these tetrads will go through a process called crossing over, where a piece of non-sister chromatid will exchange places with the identical size piece of another non-sister chromatid. So we exchange genetic material. So remember, this was a whole pair of sister chromatids that is homologous to this pair of sister chromatids. They will come together and sit on top of each other. They're not drawing them sitting on top of each other because then it makes it hard to see what's happening. But what will happen is, remember that these chromosomes are made up of DNA polymers, just nucleotide connected to another nucleotide, connected to another nucleotide, and wrapped around proteins. Well, we will take a piece of, let's say, the top 5,000 nucleotides, break it off from the blue, and we'll take top exactly 5,000 nucleotides up to the exact same point from the other homologous chromosome, and we'll exchange places. There's a whole slew of different proteins that are working in order to make this done precisely. Okay. At the end, you end up with 4 chromatids that are now not identical to any of themselves. Some chromosomes, or I should say some tetrads, undergo multiple crossing over events. So here is what it would look like under a microscope, and you can see that it's like all entangling and crossing over multiple times in multiple places. In summary, during prophase 1 of meiosis, chromosomes condense, become visible under the microscope, the spindle of microtubules starts to form. The DNA that has been replicated consists of 2 sister chromatids, because that happened in S phase, so they're still there. And the homologous chromosomes, which are made up of sister chromatids, will come together and form a tetrad or a foursome. And within each tetrad, non-sister chromatids, so the ones that are not identical, will exchange pieces of themselves in order to create 4 unique chromatids. So after this, we slowly move through to metaphase 1, at which point all of the tetrads, and in humans this is 23 tetrads, will be lined up on the metaphase plate. And microtubules from opposite poles will attach to each homolog pair and not sister chromatids. The homologs are aligned at the metaphase plate side by side, and they will have a random directionality. So orientation of each pair is going to be random. And what that means is when they're at the metaphase plate in the center, some of the red ones will be facing the left pole and some of the bluish ones will be facing the red pole. And then for the next tetrad, it may be the opposite. So when they get split apart, you get some of each combination going to separate poles. In mitosis, none of this was even relevant because we didn't have tetrads. We had two identical sister chromatids. So it didn't matter if this was on the left and this was on the right or vice versa, because they were exactly identical. So when they get split apart during anaphase of mitosis in the previous chapter, you just get one of each identical copies going to each pole, and there's nothing to keep track of. But here we have 4 genetically unique chromatids. So depending on how they line up, like which one is connected to each pole, they will get separated in different ways that mixes them up. If a cell only had 2 tetrads, as is shown here, they can be lined up in 2 possible ways and therefore get split up in 2 different ways. If we have 3 tetrads, if there are 3 tetrads present in the cell, then there are 8 different ways that they can split up. But in humans, if there are 23 tetrads, and try to go back and remember why there are 23 tetrads in a human cell in this process. In humans, there are 23 tetrads at this point in the process, and depending on how they line up at the metaphase plate, you get 8,388,608 different combinations of which chromosomes can be facing which way. So here are 8 possibilities. In humans, there are almost 8.5 million possibilities of how the DNA can get split up at this point. Okay. So in metaphase 1, pairs of homologs align along the metaphase plate, and then in anaphase 1, microtubules of the spindle shorten and homologous pairs are separated. The sister chromatids still remain attached to one another at this point. Each pole has a complete haploid cell of chromosomes, but they're each consisting of sister chromatids. So this is a strange kind of transition phase and is not a normal cell arrangement. We have just gone through independent assortment of maternal and paternal chromosomes. That refers to the different orientation of tetrads at metaphase plate of metaphase 1. Okay, so anaphase, homologs split away from each other, sisters stay attached. During telophase, nuclear envelope somewhat reforms, and you have these transition cells. Sister chromatids are still attached to one another, but they are no longer identical because of that crossing over and recombination of DNA that happened in prophase 1. Cytokinesis may or may not occur after telophase, it depends. And meiosis goes right after the end of telophase 1, although there could be a pause, but no other events take place between telophase 1 and meiosis 2. In meiosis 2, the division very closely resembles that you learned in mitosis from the previous chapter. So in prophase 2, nuclear envelopes dissolve and new spindle apparatus forms. Metaphase 2 chromosomes align at the metaphase plate. Anaphase 2 sister chromatids are now finally separated from one another. And in telophase 2, nuclear envelopes reforms and you have 4 daughter cells. Each has haploid number of chromosomes, and all the 4 cells are genetically unique in comparison to one another. Okay. So meiosis 2 does not have any unique features of the process. Sisters separate, 4 new cells form, but the result is unique. There are 4 cells containing haploid sets of chromosomes, 4 genetically unique cells that are each haploid. And in animals like ourselves, these are the resulting eggs and sperm, where in plants, fungi, many protists, like one sperm can make more sperm just by mitosis. So from one sperm you can manufacture lots of sperm in other organisms. But in animals, like humans, that never happens. New sperm or new eggs have to be made directly from germline cells. So yeah, there is a limited amount that can be manufactured. Females will generally put all of the energy into one of the 4 resulting cells. So one of them will be meant to be a fully functional, fully stocked egg with lots of mitochondria, backup energy stored, molecules with energy stored in them, all the endoplasmic reticulum and Golgi and everything else that's necessary, while the other 3 cells would have been just a receptacle for the division of chromosomes and will be recycled, broken down. Okay, so normally every month, a human female will release only 1 egg, although the process of meiosis normally results in 4 cells. So on the exam, I do want you to just say that the result of meiosis is 4 genetically unique haploid cells. Okay? Even though practically in human females, there's an extra process that results eventually in just 1 egg per month after each of the cycles. But that is not going to be on the exam, and it's just kind of a interest piece that you might be asking about. This process is incredibly complex, of all this division, lining everything up, duplicating everything, breaking pieces off. It requires so much energy, huge amount of energy, a lot of different proteins that are working as enzymes and in other capacities in order to make this all go smoothly. And much of the time it does happen very smoothly, but it's not perfect and mistakes are made. Okay. And the errors that I want to mention are most of them result in non-disjunction, where the chromosomes, when they're supposed to split apart, just don't split. So remember, they split apart twice. In anaphase 1, homologous chromosomes that were attached in a tetrad split away from each other to result in just the sister chromatids being attached. In anaphase 2, it is the sister chromatids that are split apart to be full-fledged, just individual single chromosomes. If either of those times chromosomes that are supposed to separate do not separate, then you have what's called non-disjunction, or failure to separate and move to opposite poles. Okay. This is the cause of most miscarriages, which can happen even before a female realizes that she may have been pregnant. Many also happen during the first 12 weeks of pregnancy and can happen much later as well. If non-disjunction occurs during meiosis 1, then the homologs do not separate and they're just stuck to each other. So you have a tetrad that ends up going, the entire tetrad going to one pole and none of that tetrad going to the other pole. So when these cells continue into meiosis 2 and then sisters do separate appropriately, the resulting gametes will all have abnormal number of chromosomes. 2 of them will have an extra chromosome and 2 of them will have missing chromosomes. So this is problematic. Not having all the necessary DNA will generally result in a non-viable zygote. Having extra DNA may result in a miscarriage or a spontaneous abortion, or it is very infrequently, but it may survive. For example, in Down syndrome, which is also called trisomy 21, the 21st chromosome is the one that was in non-disjunction. So if this tetrad happened to be tetrad of chromosome 21, so maternal chromosome 21, paternal chromosome 21, and each is made up of sister chromatids, and all 4 of them went to one side, eventually 2 of them end up in the same egg. So egg now has 1 of each chromosome, except it has 2 copies of chromosome 21. When this egg gets fertilized by a sperm that has 1 of each chromosome, then the resulting zygote will have 3 chromosomes number 21. And that may develop into a person who has Down syndrome, which is also called trisomy 21, which has some health conditions. They do become the cutest kids and the sweetest people, but they have some increased rates of heart disease and other possible conditions, and there's a range. If non-disjunction happens in meiosis 2, that means that during the first division, or anaphase 1, the homologs separated just fine and exactly as planned. But during the second round of division, in meiosis 2, when sister chromatids were supposed to separate during anaphase 2, one of the sets didn't get separated. So maybe this protein didn't get attached correctly, or something went wrong with this spindle here. In that case, 2 of the resulting gametes, so 2 out of the 4, will have the expected number of chromosomes, 1 of each, while the 2 other gametes will each have +1 or missing 1 chromosome. So again, if this happens to be chromosome number 21, and this egg becomes fertilized by a sperm with 1 set of each chromosome, the resulting baby will have trisomy 21. They'll have 3 copies of chromosome 21.